Hello there and welcome to Botany 101. This is going to be a review session for Exam 2. My name is Angela Nishimoto and I'll lead you through this. Okay, so today we're doing the Exam 2 review. Okay? So for one thing, flowering plants number more than 250,000 species. These are flowering plants, also known as angiosperms, also known as magnolia phyta, also known as anthophyta. So for the flowering plants, there are 6 to 11 species of flowering plant that provides 80% of the food of the world, of all the humans. Okay, 6 to 11 species provide about 80% of the world's food. Okay, we'll come upon this later on in our little talk. Okay, so flower structure. Okay, because without flowers, you don't have flowering plants, right? So the flower, of course, is a sex organ of the flowering plant. Okay, and it was called a short shoot with modified leaves. Okay, so all the parts of the flower is modified leaves. And Goethe, the famous 19th century German romantic philosopher, poet, and scientist, said, "Alles ist Blatt." That means it's all leaf. Okay, so all the parts of the flower, which will come up next, okay, are all made of modified leaves. Okay, it's a short shoot with modified leaves. So the outermost part of the flower are the parts that oftentimes are the most leaf-like part. Okay, they can be green, they can be other colors as well, but sometimes they're green. And all the sepals, which is what it's called, all together are called the calyx. Okay, and the calyx functions in protecting the rest of the flower when the flower is in its bud form. Okay, that's the function of the sepals and the calyx, is protection. Interior to the calyx or the sepals, you have the petals. Okay, and they're usually the colored part of the flower. They could be any color you can think of. Okay, from white to blush, all the way to brown and purple and black, with blues and all the colors in between. Okay, so this particular little image here has pink petals. Okay, all the petals together are called the corolla. All right, and the petals all function in attracting a pollinator. So the sepals function in protection, the petals function in attraction. Okay. Now interior to the petals, you have the stamens. Okay, stamens. These are the male part of the flower. So remember, stamens. And all the stamens together are known as the andrecium. Okay, that just means the male parts. Okay, they make the pollen. Okay, and the stamens are made up of an anther part. That's the part that produces the pollen, and it also has a filament. Okay, and the filament connects the anther to the rest of the flower. Right? Now the fourth part of a complete and perfect flower is this part here. Okay, it's a female part of the flower, which is what gynecium means. So the pistil can be made of one or more carpels. Okay, a carpel is a folded over leaf that has seeds inside. So this one has a single carpel with a single ovule in it, which would become the seed after pollination and fertilization. Okay, so the pistil is known as a gynecium, or the female parts. Okay, and the pistil is made of the stigma, which is here, this receptor surface here, the style, this tube here. Okay, so the sticky stigma, the stylus style, and then we have the ovary that has one or more ovules in it. Okay, so those are the parts of the pistil. Okay, remember the pistil is a female part of the flower. Okay, don't get it confused with the male part of the flower. Okay, now fruits. In 1893, the U.S. Supreme Court stated that the tomato is legally a vegetable. The part that we eat is legally a vegetable. Okay, but actually tomato is botanically a fruit. They could say that the tomato was legally a vegetable because the tomato actually is not sweet and generally is not used for desserts, whereas I guess the Supreme Court defined a fruit as something that's sweet and something that has its place in desserts, such as pies, tarts, things like that. Okay? So the tomato is actually botanically a fruit. It's a mature ovary with other parts. Okay, a mature ovary with other parts, especially seeds. Like as you can see in this here, section tomato. Okay, that's what a fruit is, a mature ovary with other parts, including seeds. Okay. Now, so how can we differentiate the vegetables from the fruits? Okay. So vegetables generally are made of the vegetative organs of fruits, like the leaf, okay? In lettuce, cabbage, and onion, the leaf is a part of these different kinds of plants that we eat, okay? The petiole we eat in celery, that's actually the leaf stem, right? You remember the blade and the petiole are two parts of a simple leaf, okay? So a petiole is celery, okay? A stem that's used for food is like the Irish potato, because remember the Irish potato is a special kind of stem known as a tuber that stores starch. It grows at the end of a rhizome, okay? And then we have roots, such as storage roots, such as beet, carrot, and sweet potato. Okay, so these are different kinds of vegetables. Vegetables can also be made of flowers, okay, as in the case of broccoli and cauliflower. All right. And flower buds include the artichoke, okay? But fruits develop from flowers, okay? So fruits only occur in flowering plants. They do not occur in gymnosperms. They do not occur in ferns. They do not occur with a fern ally such as Psilotum or Equisetum or Lycopodium or Selaginella. Okay, they do not occur in mosses. So fruits occur only in flowering plants. Okay? Okay, now we're going to get into different kind of fruit types, and especially the fleshy fruits. That's what we're going to focus on. Okay? So a drupe, okay, is a single-seeded fruit, and the seed has a hard casing over it, such as what we see in peach and olive and lychee, as well as mango, nectarine, plum. These are all different kinds of drupes. Okay? And berries are thin-skinned fruits with a few to many seeds. So examples of berries include kiwi fruit, tomato, guava. Okay? And we have another kind of fruit called a pepo. It's a type of berry, but it's a special type of berry that has a rind, a thick rind. Okay? So some examples of pepos are watermelon, cantaloupe, and cucumber. Okay? Another kind of berry is a hesperidium, and hesperidiums are the citrus fruits, okay, such as oranges, lemons, grapefruits, jabong, kumquat, okay, limes. Those are all examples of hesperidiums. Okay, you'll have to know these. Okay? And palm fruit are apples and pears. Okay? So remember that apples and pears are not berries, even though they do have a few to several seeds. But this part here in the apple is where you find the seed, and that part is the ovary. Whereas other kinds of simple fruits that we see, okay, we actually eat the ovary part, but this part we eat the receptacle tissue, like this part here in this apple, or this part here in this pear. Okay, so apples and pears are actually palm fruits. Okay. Now, aggregate fruits include raspberries and strawberries. These are fruits that develop from a single flower that has many pistils. Each of these little fruitlets on this raspberry is actually an individual fruit. an individual pistil. Okay, and all of these little so-called seeds on the surface of the strawberry are actually individual ovaries. Okay. So each of these is actually a fruit called an achene, and it's on a swollen part known as the receptacle tissue of a strawberry, and that's the part, of course, that we like to eat. Now, multiple fruits come from multiple flowers. Okay, so that includes pineapple. So each of these little lozenge shapes over here on the surface of the pineapple fruit actually represents a flower. And in the fig, the flowers are internal to the outer part of the fig, the outer skin of the fig. So each one of these little drupelets is actually an individual fruit. So that comes from an individual flower. Okay, so multiple fruits come from multiple flowers. Okay, another example of a multiple fruit is noni. Another example of the multiple fruit is a ulu, or breadfruit. Another example of a multiple fruit is the hala, the female hala flowers that mature into fruits. Okay, that's called a cirrhosis. Okay. Now, dispersal of fruits and seeds. You can have wind dispersal, as in the cases of dandelions and orchids, or you could have dispersal by water, such as some sedges and also the naupakakai. Okay. And also you have different modes of animal dispersal. Okay. So animals can actually have seeds or fruits stuck onto their muddy feet, like sedges often get moved thousands of miles from one place to another by way of muddy feet. They think probably that ahuava probably came that way here from Southeast Asia. Okay. Or another means of animal dispersal is a fruit or a seed could stick to fur or feathers, such as we see in the cocklebur. You see all these little curved things here. They're like Velcro. They make the cocklebur stick onto your pants leg or onto the fur of a dog or onto feathers of a bird. Okay. The third means by which animals can actually move fruits or seeds is that they can eat it and then excrete them with a little drop of fertilizer there. Okay, so some examples of fruits that are eaten and excreted include the blueberry. Look at these beautiful colored fruits here, and also the ohelo, okay, which is native to Hawaii. Okay. Now we're getting into physiology here. Okay, so osmosis has to do with a solvent and a solute. Okay, so a solvent is something like water that dissolves a solute or some kind of solid, such as table salt or sugar, and the solvent and the solute together make up a solution. Okay. So in this case, we have water as a solvent. Okay. So osmosis itself is the movement of water through a semi-permeable membrane from an area of higher concentration to an area of lower concentration. Okay, it's a passive process that doesn't need an input of energy to make it happen, because the universe wants everything to be balanced, so everything kind of moves toward equilibrium, including the concentrations of solvent and solute in and out of the cell. Okay, so water in the cell moves by osmosis from cell to cell, and in a plant cell, the cell wants to be turgid or full of water. Turgid means full of water. Okay, so you might have experience with this, like when you go to, say, a store and you get these pre-cut carrots and celery. If you soak them in water, they get more crunchier, right? They get more full of water, and they get more kind of tasty to us because they're full of water, and they're more pleasurable to eat when they're crunchy. So soaking carrots and celery can actually let that come about. Okay. So now, how does water actually travel 300 feet up from the roots of a 300-foot tree up into its leaves that are 300 feet above the ground? Okay. First of all, you had Stephen Hales who talked about root pressure. Okay. So if you prune a plant, water exudes from it. Okay. So that's something that he remarked upon. Okay. So he also remarked upon the pulling force from evaporation from the leaves. So if you recall, water molecules are polar, right? You have the oxygen side of the water molecule pulling electrons to itself, because oxygen is among the most electronegative of all the elements, right? So they have a tendency to pull a negative charge to themselves, or electrons. Okay, so one side of the water molecule is slightly negatively charged. The other side of the water molecule is slightly positively charged. Okay, so water molecules are polar. Okay, so because they're polar, they stick together. The negatively charged part of a water molecule will be attracted by hydrogen bonding to the positive parts of another water molecule. Okay, so they stick together. Another thing is you have adhesion, where it sticks to the walls of the xylem. Okay, so cohesion, you see these water molecules sticking in hydrogen bonding to each other, and you see adhesion where they're sticking to the side of these walls here, okay, by adhesion. Okay, sticking to something different. Cohesion, when like things stick together like water molecules. Adhesion is like when water molecules stick to the side of a glass graduated cylinder or test tube. You have this meniscus, right? That's because the water molecules are sticking to the side of the glass. Okay, it does the same to the xylem walls, right? And water evaporates, okay? So water evaporates out of a plant by diffusion out of the stomata, okay? So the diffusion happens without a membrane, because of course the water evaporates through the stomata, and there's no membrane separating the opening of the stomata from the outside atmosphere. And you have diffusion happening outside of the stomata because the air has less water in it than inside the plant does. So water actually moves out of the plant through diffusion from an area of higher concentration inside of the plant to an area of lower concentration outside of the plant. Okay, so this diffusion creates a pull on water from lower down. Okay, so what you have here is that you have the water leaving the stomate. Okay, water being drawn through the vascular bundles here, out through the spongy layer and out through the stomate. Okay, and that creates a pull on the water further down in the stem, which is pulled up by cohesion and adhesion, where the water molecules are sticking together, creating a whole columns of water molecules moving upward, okay, then into the leaf, okay, and that creates a pull of water from the root, and it actually enters the root hair, okay. So you have the force of cohesion and adhesion actually allowing the water molecules to be pulled upwards, and evaporation actually is a driving force in here. So the transpiration or evaporative loss of water through the stomates actually pulls up the water through the stem and from the root into the stem and into the leaf. Okay? So the roots take water from the soil. So how do they do that? Remember, so roots, okay, root cells have a cell membrane, right? So roots take water from the soil through osmosis, okay? So what happens is that generally the soil has more water in it than the root does, so the water passively moves from the soil into the root because the root has less water in it because the water is being pulled up through the stem into the leaves and out the leaves to the stomate. So that creates a pull of water. So that actually allows for the global cycling of water through trees and through other plants. Okay. Now stomates have two guard cells. A stomate is a pore through which gas exchange can occur, and this is a stomate. So it looks a little like a mouth, right? So stoma means mouth, okay? So the pore is called the stomate, and you have two guard cells, okay, that actually guard the entrance to the stomate, okay? And here you can see some chloroplasts, because generally the stomates are on the surfaces of the leaves, and they're just about the only cells on the surface of the leaf or in the epidermis that has any chloroplasts in them, okay, because they need the chloroplasts to actually harvest light energy to do it, okay? So stoma will actually, when it's full of water, the guard cells are turgid and the stomate opens, okay? When the cell loses water, then the guard cells lose turgidity or gets flaccid and the stomate close, okay? And like I said, it regulates gas exchange, and the stomate also regulates transpiration, and they close during water stress, okay? So that's the stomates and the guard cells. Now metabolism. Okay, metabolism is the sum of all chemical reactions in an organism. Okay, so generally the sum of all the chemical reactions in an organism requires energy. Okay, not just that, is that living things are the only, like non-living things do not have metabolism, but living things do. Okay, we call that energy is the ability to do work. Okay, so here, kind of showing the sun, showing the leaf that it's shining on. Okay, so the sun is actually doing work by warming and also sending out its electromagnetic energy in the form of waves. Okay, also in packets known as photons. Okay, that's the work it's doing, and the energy that the plant actually absorbs is used to make energy in a chemical way. So it absorbs radiant energy from the sun and converts it into chemical energy. Okay? Right, so there's a guy running across the screen there. Okay, so now we come to photosynthesis. Okay? So oil and coal, 90% of it is actually used in transportation, communications, electricity for household and uses by businesses, and also generally that's energy from the sun. Because if you can recall from before when we had lecture, okay, that the oil and coal are actually fossil fuels, right? They're actually fossil fuels from plants and algae mostly that had lived hundreds or tens of millions to hundreds of millions of years ago, and the energy ultimately came from the sun. Okay, so remember that the only kinds of organisms that can do photosynthesis include plants, algae, and cyanobacteria, which are also known as blue-green algae or blue-green bacteria. Okay, so plants, algae, and cyanobacteria are the only organisms that can do photosynthesis. Okay, and that keeps all the rest of us, the rest of us critters alive. Okay, so photosynthesis is the sole means of sustaining life. Okay, photosynthesis actually means to actually make something with light. Okay, so a photosynthate is used, that's actually the product of photosynthesis. It's used to make wood for the plant, fibers in the plant, and other structures in the plant. It also goes into making sucrose, which is the form of sugar that's moved in the plant from one place to another, and starch, which is a storage product of plants. It also goes into making amino acids, of course, which of course are the units that form proteins. Okay? So 40 to 50% of the photosynthesis that goes on our planet happens in water through the photosynthesis of microalgae and macroalgae. Microalgae are just single-celled algae such as diatoms and dinoflagellates, okay, and other algae that are single-celled, and the macroalgae are the ones that have multiple cells making up their bodies. Okay, here's a look at the vast oceans on our planet. Okay, so there are probably, what, a zillion. That's just a made-up word, but there are like uncountable number of microalgae in these oceans all over the world. Okay. Now the equation for photosynthesis. Okay. 6 carbon dioxides plus 12 waters plus light. Okay, here. So here's the carbon dioxide during the Calvin cycle. Here's the water during the light reactions. Okay. And then what you end up with, and here's the light, what you end up with is sugar, C6H12O6, sugar, oxygen gas, and you also end up with some water too coming out. Okay? So that's the equation I want you to know. 6 CO2 plus 12 H2O plus light goes to C6H12O6 plus 6 O2 plus 6 H2O. Okay? That's a balanced overall chemical equation for photosynthesis. And photosynthesis, of course, happens in 2 phases. You have the light reactions and the Calvin cycle. Okay? So light acts both as a wave and also acts as a particle. Okay, both. All right. And light is a visible light, is part of the electromagnetic energy, part of the electromagnetic spectra. Okay. So the longer the wavelength, the lower the energy. So the visible light has the longest wavelength, is red. Okay. Red has the longest wavelength of visible light and has the lowest energy. And violet has a shorter wavelength, or the longest wavelength of visible light, and has higher energy. So below red, there's, let's see, infrared, right? And above violet, there's ultraviolet. And of course, those are not visible to us. Okay? Infrared is what's responsible for heat energy that we feel, like when we're in the sunshine or under lights. Okay? And the ultraviolet is like UV light, yeah, which can be can damage. It can be damaging radiation. So the higher the energy, the more damaging it can be. So the part above the visible spectrum can be like X-rays or gamma rays can be very damaging to your body. Okay, so here's looking at the spectra. Okay, so red is a longer wavelength. Here's orange, so it's from 750, 780, I think. Then you have here orange, then yellow, then green, then blue, then violet. Okay. Okay, so about that range. Okay, so yeah, violet to blue and red orange to red is a wavelength that plants actually absorb. So why do leaves look green to us? Why do leaves look green to us? Because it's absorbing in the violet to blue and the red to orange to red. Okay, so and it's reflecting and transmitting green. That's why leaves look green to us. Okay. Now chlorophyll is the main pigment in photosynthesis, but there are other pigments that also come into play. It allows for a broader absorptance spectrum. So they include the carotenoids, and carotenoids include the carotenes that are orange to red, usually. The xanthophylls that are yellow to brown. Okay. And there are other water-soluble pigments called anthocyanins. Okay. Anthocyanins are water-soluble pigments that kind of range from pink to red to purple to blue. Okay. And they're oftentimes responsible for the color that you see in flowers or things like red onion or red cabbage. Okay. And then we also have phycobilins that are blue and red that are found in blue-green algae or cyanobacteria, and also red algae. Okay. So phyco, remember, means alga, right? So these phycobilins, you can have phycoerythrin and phycocyanin. Okay, so those are reddish pigments and also bluish pigments, and they're found in red algae, and they're also found in blue-green algae or blue-green bacteria or cyanobacteria. Okay. Okay, now we also have chlorophyll C, chlorophyll D, chlorophyll E. Okay, which are variations on different kind of chlorophylls, and these are found in algae. Okay. Now the light reactions of photosynthesis happens in the grana of the chloroplast. Okay. So remember, the grana of the chloroplast is made of the thylakoid membranes, all stacked like coins. Okay. So first of all, the photon hits the chlorophyll. Okay. Water is split. Okay. Water releases electrons and hydrogen ions. Okay. And also releases oxygen gas. Okay, so water is split, breaking it down into its component electrons, hydrogen ions, and oxygen. Okay, so the electrons are then passed along an electron transport system. Okay. Okay, here you have the electron transport system. Okay, so we have water, okay, which comes in, split by light, okay, oxygen gas being given off, hydrogen ions being generated, and you have here electrons being excited in this photosystem 2, okay. So the electrons are then passed from one electron acceptor to the next electron acceptor, okay. And when that's happening, what's actually happening is that provides energy with which to pump hydrogen ions across the concentration gradient to inside of the thylakoid lumen, okay. So, and that actually allows for the hydrogen ions to diffuse back through an ATP synthase protein enzyme, okay, and that actually makes ATP, okay. Now that's photosystem 2. Now in photosystem 1, okay, light hits a pigment molecule, and that actually bounces around until it actually raises the electron to a higher energy state. Okay, then it's also passed down through electron acceptors and is used in the formation of NADPH, okay, which is an electron carrier plus a hydrogen carrier. Okay, NADP plus the hydrogen ions goes to NADPH. Okay. Anyway, so two products of the light reactions are ATP and NADPH. Okay. So ATP is produced, okay, as we saw that image here. The hydrogen ions diffuse out back to the ATP synthase, okay, and produce ATP. Okay, reduced NADPH, or reduced nicotinamide adenine dinucleotide phosphate, NADPH is formed. Okay, that happens here. Okay, where you have the NADP plus being the electron and the hydrogen being passed to it, and that actually generates NADPH, which is an electron carrier. Okay. Now the Calvin cycle is the next part. So the products of the light reactions are ATP and NADPH. Okay. So the Calvin cycle happens in the stroma. Okay, it uses ATP and NADPH that was provided for it by the light reactions, okay, and it uses electrons, okay. So what happens is that carbon dioxide comes in during the Calvin cycle. Okay, remember it from the formula CO2, right? So carbon dioxide combines with ribulose bisphosphate, okay, and is converted to sugars eventually after going through some changes. And some of this sugars are recycled, okay, in the form of 3-PGA phosphoglyceric acid, okay. So some grasses and dry area plants fix carbon differently, okay. Anyway, so what we have here is looking at ribulose 6-phosphate. Okay, so what happens is, let's see, so here. So carbon dioxide comes in and joins to ribulose bisphosphate. Okay, and then what happens is that breaks down into 2 or 3 carbon fragments. Okay, and eventually you have the formation of glyceraldehyde 3-phosphate, okay, and that, most of that, generally for every 12, 10 of them are used to recycle the RUBP, okay, and only 2 are actually used to come together to make glucose, okay. And here you have this enzyme called rubisco that helps facilitate the whole thing, that helps the carbon dioxide come together with the ribulose bisphosphate, okay. Okay, so carbon dioxide is fixed to make carbohydrate, okay. So this Calvin cycle is the main way that carbon enters the web of life, okay. So you have 6 carbon dioxide plus 6 ribulose bisphosphates, okay, with help from Rubisco. Okay, so then you have the 6 carbon complexes splitting into these 3 carbon 3-phosphoglyceraldehyde or phosphoglyceric acid. Okay, and so you have NADPH and ATP giving energy and electrons to reduce that to glyceraldehyde 3-phosphate. Okay, what we see here. Okay, ATP and NADPH. Okay. Anyway, so you have 10 GA3Ps, glyceraldehyde 3-phosphates, plus 6 ATP, makes 6 ribulose bisphosphates, or RUBPs. Okay, so you have 2 glyceraldehyde 3-phosphates net. Okay, and that's actually used to go off to actually form sugars. Okay. So they're called C3 plants since Rubisco forms 3-carbon phosphoglyceric acid. Okay. So Rubisco can fix both carbon dioxide and oxygen gas, and that's an oxygenase activity called photorespiration. But we're not really going to talk about that very much at all. Okay. Okay, so the light reaction in the grana, the photon hits the chlorophyll, the water is split, releases electrons and hydrogen ions, and also releases oxygen gas, as I just explained. Electrons are passed along an electron transport system. Okay, like we saw before. Photosystem 2, electrons are passed. Okay, and what they do is they'll pump protons into the thylakoid lumen. All right. And then the protons will then diffuse out through the ATP synthase. And produce ATP. And then NADPH is formed by a similar action where you have light hitting photosystem 1. Okay. And the electron being excited and passed down an electron transport chain, and the energy is actually used, and the electron is used to form NADPH. Okay, so photosystem 2, photosystem 1, that's just kind of showing their their peak absorption spectra. Okay, so you're showing photosystem, let's see, photosystem 2 is first. Okay, you have the energy transfer, excited electron. Okay, actually, so that allows for the formation of ATP eventually. Okay, you have another ray of sunshine hitting photosystem 1 that also excites electrons, and then that's going to be passed down electron transport system. Okay, just a different way of looking at it. Okay, so the water splitting photosystem, photons are absorbed. Okay, the electrons get excited and are passed on to electron acceptors. Okay, water is split to form oxygen and 4 hydrogen ions and 4 electrons. Okay, now electron flow and photophosphorylation. Okay, so electrons are passed down an electron transport system, and hydrogen ions are moved across the membrane. ATP is formed from ADP, adenosine diphosphate, plus a phosphate group. Okay, now we also have the NADPH generating photosystem. Okay, where photons are absorbed by P700, electrons get excited, and they're transferred from one electron accepted to another, and what that would eventually do is reduces NADP plus to NADPH. Okay, that's what we see here, as we've seen before. Okay, now chemiosmosis, okay, happens inside the thylakoid membrane. Water is split, producing what? Okay, think about it. Okay, ask yourself that question and see if you can answer it. Okay, then electrons go through the electron transport system. Okay, hydrogen ions move to the inside of the thylakoid membrane and they accumulate. So hydrogen ions then move through the ATP synthase or ATPase and produces ATP. Just what we saw before. Okay, so we have the photosystem 1, photosystem 2, light being absorbed and the electron getting excited, being passed down electron transport system. That powers the pumping of the protons to the inside of the thylakoid lumen. Okay, so you have a high concentration of hydrogen ions inside the thylakoid and a low concentration of hydrogen ions outside the thylakoid. And whenever you have that kind of concentration gradient, you will have potential energy. Okay, so then what happens is the hydrogen ions then diffuse to the ATPase or the ATP synthase and produces ATP. Okay, then the second part, you have light hitting P700, photosystem 1, exciting the electron, and the electron plus a hydrogen ion coming together with NADP plus, forming NADPH. Okay, so NADPH is an electron carrier as well as a hydrogen ion carrier. Okay, now, so that does us for photosynthesis. Okay, that was a lot. Okay, I know it is. Okay, don't be discouraged. Okay, we'll just kind of soldier on. Okay, now aerobic respiration. Okay, you need oxygen gas for aerobic respiration. Okay, the equation is sort of like the inverse of photosynthesis. You have C6H12O6 plus 6O2 going to 6CO2 plus 6H2O plus energy. Okay. It's actually showing plants, algae, some bacteria doing photosynthesis. They're known as autotrophs, actually doing photosynthesis, taking in carbon dioxide, okay, and water, okay, and then releasing oxygen, which is used by fungi and animals, like this young woman here. She looks like a person. Okay, so what animals will do, like us, is that we take oxygen and we use it to break down food in cellular respiration. Plants do this too, by the way. Plants also do cellular respiration. Here's a praying mantis. Okay. Okay, so what you have here is that you have anaerobic respiration and fermentation happens without oxygen gas. Okay. So what happens is that you have C6H12O6 going to 2C2H5OH plus 2CO2 plus energy. Okay, it's actually fermentation. Okay, okay, so we have that. Okay, so either pathway, you can actually either go lactic acid fermentation or you can go through alcoholic fermentation. Okay. So alcoholic fermentation was the first one here, and the second one here is lactic acid fermentation. Okay, so both of them are anaerobic, and both of them actually lead to the production of 2-ATP, which is kind of like a low turnout for ATP, but there are many organisms that actually go to an anaerobic pathway. Okay, when aerobic pathway is not necessary or not available to them, or when oxygen is not available. Say, for example, if you're sort of like not in good shape, sort of like I am not in good shape right now, okay, and what you did is you decided, okay, so tomorrow I'm going to go and jog up the hill, you know, and then you're going to be jogging and you're going to be sort of getting tired and breathing hard, because your heart and your lungs are actually doing their best to keep your blood supplied with oxygen, because oxygen is actually being taken to your cells, which used to break down the sugar in your cells to ATP for the ready energy that allows your muscle contraction as you're running Okay, so if you're running and you're not in condition, sooner or later your body's going to switch to the anaerobic pathway. You're going to go into lactic acid fermentation, which only allows 2 ATP. We're going to find out later that aerobic respiration actually allows for more ATP to be formed. Okay? Okay, so aerobic respiration includes glycolysis. Okay, so aerobic respiration happens in 3 parts. You have glycolysis that happens in the cytoplasm. You have the Krebs cycle that happens in the mitochondrial matrix, and you have the electron transport system that happens in the inner membrane of the mitochondria. Okay, so in glycolysis, the overall reaction is that glucose, well, first of all, glucose is converted to a fructose with 2 phosphate groups. Okay, the fructose splits into 2 3-carbon glyceraldehyde 3-phosphates. Okay, then hydrogen, energy, and water is removed to form pyruvic acid. Okay, so the overall thing that happens in glycolysis is you have a 6-carbon glucose that eventually forms 2 pyruvates or 2 pyruvic acids. Okay, so that's actually looked at here. So glucose here, okay, is 2 ATP are actually invested into making the system start. Okay, so first you have 1 phosphate group joined to it, then you have another phosphate group joined to it eventually. So you have 2 phosphate groups, and then what you have is a breaking down of the 6-carbon group with 2 phosphates. Okay, and then what you end up with is you end up with 4 ATP that's actually harvested. After that, water is actually given off. Okay, and you get NADH. Okay, which is the electron carrier in aerobic respiration. Okay, or in glycolysis too. All right, so don't confuse NADH with NADPH. Okay, NADPH comes into play with photosynthesis, and NADH comes into play with respiration. Okay, so the cell actually grosses 4 ATP, but it only nets 2 because it had invested 2 at the beginning. Okay. Now the citric acid cycle is also known as the Krebs cycle, and that's what I call it. I call it the Krebs cycle. It happens in the matrix, or the inner part of the mitochondrion, and it was actually delineated by Hans Krebs in the 1950s. Okay, so before, what happens before the Krebs cycle is that carbon dioxide is released from the pyruvic acid, so then what you have is a 2-carbon acetyl group, okay, that combines with coenzyme A, okay, to form acetyl coenzyme A. Okay, so here is actually looking at the citric acid cycle, or the Krebs cycle in the mitochondrion. Okay, so here you have coenzyme A being given off, and it's going to enter into the Krebs cycle here. Okay, so a cyclical pathway, just like the Calvin cycle, is that what it does, it regenerates the same molecule that it starts with, which in this case is oxaloacetate. Okay, so what happens is that the coenzyme A goes off, a 2-carbon joins with this 4-carbon, and becomes a 6-carbon, and then it becomes a 5-carbon, then a 4-carbon, then it goes through several other changes until it regenerates oxaloacetate. Okay, another thing that happens is that you get 3 NADHs. Okay, you also get ATP. Okay, 2 ATP from this process, and also 2 carbon dioxides are given off. So during the Krebs cycle, you have the complete breakdown of what's left of the glucose molecule. Okay, because 2 carbon dioxides come from the 2 carbons that were in the acetyl group of the acetyl coenzyme A. Okay. So during the citric acid cycle, or the Krebs cycle, the electrons and hydrogens ions are removed. Okay, they form 2 electron carriers, NADH plus FADH2. Okay, also forms ATP, and the carbon dioxides are removed, yeah, just like what we just saw. Okay, so here we have NADH, carbon dioxide removed. Okay, NADH, carbon dioxide removed. Okay, FADH2 being formed, oops. And also another NADH, so 3 NADHs, 1 FADH2, 2 ATP, plus 2 carbon dioxide coming out of the Krebs cycle. Okay. Now electron transport happens in the mitochondrial inner membrane, and during then the electron carriers, NADH and FADH2, will donate electrons, okay, to electron transport systems. And ultimately, these electrons ultimately fall to oxygen. Okay, remember oxygen is electronegative, right? So it has a tendency to pull electrons toward itself. So generally what the oxygen is doing is actually pulling electrons through the electron system until it falls to oxygen, okay? Also here, we also have another proton pump. Protons are pumped, forming a gradient similar to what we saw in photosynthesis, but this is happening in the mitochondrion, okay? Then other proteins actually allow hydrogen ions to go back into the matrix, okay, forming ATP, okay? So you have 2 ATP, okay, from glycolysis, you have 2 ATP from the Krebs cycle, and you'll have 32 ATP from electron transport, okay? So generally, on average, the net ATP yield for aerobic respiration is 36 ATP, okay? So here we have the citric acid cycle seen once again. Okay, so here we have pyruvate, the end product of glycolysis, okay? You see carbon dioxide being given off. So you have a 2-carbon group joining to the coenzyme A, okay? So that gives off the coenzyme A and joins with oxaloacetate. Then you have a 6-carbon compound, a 5-carbon compound, here, 4-carbon compound, 4-carbon compound, until they regenerate oxaloacetate, which is a 4-carbon compound too. Okay? You don't have to know all these steps or know all these drawings or anything like that. I just want you to get the general overall idea of what's going on here. Okay? So what you have is a generation of NADH, okay, 3 of them. You have the generation of ATP, okay, 2 of them, okay. You have the generation of FADH2. Okay, well, let's see if I can find it. Let's see. Okay. Anyway, and you also have carbon dioxide being given off. So those are the products of the Krebs cycle. So the Krebs cycle has to do with the complete breakdown of what was once a glucose molecule, right? Okay. So in anaerobic respiration and fermentation, we have glycolysis only, okay? That happens when there's not enough oxygen to complete the aerobic pathway, okay? So the hydrogen is transferred back to pyruvic acid to make ethanol or lactic acid. So either alcoholic fermentation or lactic acid fermentation, okay? Just kind of showing. So you have C6, okay, you have NADH. So what it does is it uses up all the NAD pluses. It also makes 2 ATP, okay. So what this allows in terms of lactic acid fermentation allows the regeneration of NAD plus, okay, and also alcoholic fermentation allows the regeneration of NAD plus, so then it goes back to glycolysis and do it all over again. Okay, so remember that anaerobic respiration and fermentation yields only 2 ATP, okay? Okay, glycolysis re-examined, right? So we have phosphorylation. So 6 carbon sugars receive phosphates. ATP starts the process. Sugar cleavage, it splits the 6 carbon fructose into 2 3-carbon glyceraldehyde 3 phosphates. Pyruvic acid is formed, and you have the oxidation of 2 fragments. Okay, showing glycolysis. I'm not showing you these pictures to scare you. Okay, I just kind of want you just to see that it's a complicated process. Okay, so don't need to worry about these pictures too much, but they're used to illustrate the process. So you start with glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate. So this is fructose is actually where the 2 phosphate groups are split into 2 glyceraldehyde 3-phosphates, and these are generally go on to do other things. Okay. Okay, now the transition to the Krebs cycle, you have the carbon dioxide removed, NADH produced, and the acetyl joins with coenzyme A, which we explained before. And that's actually happening here, right here. Okay, so you have the pyruvate, okay, giving off a carbon dioxide, right, and joining coenzyme A to form acetyl coenzyme A. Okay. Then the acetyl coenzyme A is joined to oxaloacetic acid or oxaloacetate, producing citric acid. Carbon dioxide is removed. Another carbon dioxide is removed. ATP is formed. And the 4-carbon compound is converted to oxaloacetate eventually. And you have some hydrogens picked up by NADH to form NADH and FADH2. Okay, that's shown here. Okay. So during electron transport, the hydrogen ions and electrons are passed along in an electron transport system, and eventually they fall to oxygen to make water. And so chemiosmosis was actually delineated in the 1960s by somebody named Peter Mitchell. Okay, so chemiosmosis. Okay, what happens? Okay, what happens is you have these concentration gradients. You kind of pump the protons to this side of the membrane, which would be on the inner part of the inner membrane of the mitochondrion. Okay, in the matrix. No, this is the matrix here. Okay, it's going to pump it to the other side. And then what it'll do is that the hydrogen ions will then diffuse back to the ATP synthase and form ATP from ADP plus a phosphate group. Okay, that's what they call chemiosmosis. Okay, so that actually happens because the electron transport systems are transporting the electrons through electron acceptors, and as the electrons are being transported, they lose a little bit of energy, and that energy is used to pump protons against their concentration gradient from an area of lower concentration to an area of higher concentration, where they'll diffuse back through these ATP synthase and produce ATP. Okay? So the hydrogen ions are pumped from the matrix to the intermembrane space, and as the electrons flow along the electron transport system in the inner membrane, the hydrogen ions diffuse back to the matrix via the ATPases, forming ATP. Okay, so a total of 36 ATP is produced after all these various goings on. Okay, so here you see electron transport system once again. Okay, now secondary metabolism. Okay, so we're on to another subject. Okay, secondary metabolism produces substances that are not essential for growth and development, but they do help plants to survive. They're responsible for certain colors or smells or poisons that plants produce. So humans use these secondary compounds for medicinal and culinary uses. They're very aromatic, many of them are. Okay, so some examples are the alkaloids, such as you see in codeine from the opium poppy, and nicotine from tobacco, okay, and quinine from the quinine tree or cinchona. Okay, so alkaloids are one kind of secondary compound, metabolic compound. We also have phenolics, okay, that include lignin from the woody plants, okay, salicin, which is from the willow tree, which is used to make aspirin, and of course the very famous tetrahydrocannabinol, that's an active principle in marijuana. Okay, so those are all phenolics. So these are secondary compounds. Remember, they don't have to do with growth or development. Okay. The third group are the terpenoids, and they include camphor, like from the camphor tree. They include menthol from the eucalyptus, and they include rubber from rubber trees. Okay. We have camphor, menthol from the eucalyptus tree, and rubber from the rubber tree. Okay. So those are the secondary metabolites. Okay. Now growth, what is it? It's an irreversible increase in mass or size due to division and enlargement of cells. Okay, so due to cell division and cell enlargement. All right. So what actually is responsible for them? Okay, some things are called hormones. All right. These are organic substances. They can either be lipids or they can be proteins that are produced in tiny amounts in one part of the plant and transported to another part of the plant where it has its effect. Okay, so we're going to do the survey of the plant hormones. So in 1881, auxins were investigated by Charles and Francis Darwin, Charles Darwin of evolution fame. Okay, so coleoptiles bend toward light. Okay, that's what they noticed. Okay, so what they did is they covered the tip, and they found that when they do cover the tip, it doesn't bend. Okay, so they figured out that there's some sort of substance that was produced at the tip that allowed the coleoptiles to bend toward the light. Coleoptiles are like the tips of germinating seedlings. Okay, so in 1926, Fritz Wendt cut coleoptiles of oats. He placed them on agar. Okay, and then put agar. Remember, agar is a substance that's gotten from red algae. It's like a gelatinous substance gotten from red algae. It's used a lot in tissue culture and in molecular biology labs. Then he put the cut oat coleoptiles, put it on agar. He put the agar on the cut coleoptiles. Okay, and what happened is that he put it on and straight up. Okay, so when he put it straight on, the coleoptiles continued to grow straight up, but when he put it off center, it tended to bend away from where it was. Okay? So the more coleoptiles they used to make the solution, the more response they got. So they were able to show that something is produced at the tips of shoots, okay, that moved into the agar. So here's some agar in a petri dish. Okay? And that substance was responsible for the bending away of the stem. Okay, so he named it auxin. Auxin means to increase. Okay, to increase. Okay. Now auxins are produced mainly in apical meristems that you see here. Okay, and in buds and all actively growing young parts of the plant. And they stimulate cell enlargement. They also trigger other hormone production. They facilitate the growth of stems, cell division in the cambium. Remember what the cambium is? It's a secondary meristem, right? It also promotes differentiation. And they can also delay fruit and leaf abscission. That means they can delay fruits and leaves falling off the plant. Okay. They also delay fruit ripening and inhibit lateral branching and stimulate root formation. And so they're used in a plant hormone known as root tone to actually allow for the growth of adventitious roots, like from a stem cutting like you see here. Okay. Auxins are also used to promote uniform flowering, as when you spray a field of pineapple plants to allow them to flower all at the same time. Okay, and they can result in seedless fruits, like of course pineapple fruits don't have seeds in them, the ones that you get from the market, right? Okay, now auxins are also used for weed killing for broadleaves. Okay, now the substance called 2,4,5-T was banned in 1979. Okay, but it was used as part of Agent Orange, which included 2,4-D and 2,4,5-T. Okay. So you see here, you see the image of the molecule. And here you see some planes, I guess, spraying Agent Orange. I think some places in Southeast Asia have never recovered from the Agent Orange that we sprayed on their lands during the Vietnam War. Okay, so Agent Orange has been held responsible for leukemia, which is a blood cancer, miscarriage, birth defect, liver and lung disease. Okay, I know somebody who died of a lung disease and he was a Vietnam veteran. Okay. Okay, I think he was like in the infantry or something, a foot soldier. Anyway, gibberellins are another kind of plant hormone, and they were discovered in 1926, almost a century ago, by Eiichi Kurosawa. And he called the disease that they caused in the rice that they grew bakanai, which means foolish seedling. Okay? He found out that this hormone came from a fungus called Gibberella fujikudoi. Okay, fujikudoi, right? And so he named it gibberellin after that particular fungus. Okay, so gibberellins come from immature seeds, root tips and shoot tips, young leaves, and also from fungi. Okay, they allow for faster growth in flowering plants. They even allow for dwarf plants to go to normal size, okay, which is something that some people might want, some people might not want. They also encourage flowering, okay. They also break dormancy of seeds, growth at lower temperatures than usual, and you can actually eliminate stratification. So stratification happens when you actually get seeds and you cold treat them. Okay, they can increase the yields of sugarcane. They actually allow for the formation of seedless grapes. Okay, for larger fruits of seedless grapes. So you know those humongous grapes you sometimes see at the supermarket? They're probably a result of the plant being sprayed with gibberellins. Okay, longer internodes to allow for the larger fruits. Okay, humongous grapes. They increase the length of the petioles of celery. We talked about celery petioles a little earlier. Okay. Okay, the third hormone is the cytokinins. Okay, so in 1913, Gottlieb Haberlandt, okay, he found a phloem chemical stimulated cell division. Okay, and initiated production of cork cambium. Okay, there's a chemical formula here, chemical form of cytokinin. Okay. In 1941, Johannes van Overbeek actually used coconut milk, or the exudate from coconut, and that's actually coconut endosperm. Okay, he used it to increase growth rate of tissues and embryos, and in fact, even nowadays, tissue culture labs use coconut water for use in terms of growing their plantlets in test tubes and in flasks and petri dishes. So in 1955, chitin was isolated from heated herring milt. Milt is like sperm or semen. Okay, so that was not naturally occurring, but they actually showed that this simple chemical could promote cell division. Okay. Then later in 1964, they found a similar substance in corn kernels, and they named them cytokinins. Okay, so they're made in root tips and germinating seeds. Okay, if auxins are present, cytokinins help to produce cell division and enlarging of cells, differentiation of tissues, the development of chlorophyll, and the stimulation of cotyledon growth. Okay, also the delay of senescence. And leaves, senescence is when leaves wither and grow old. Like it has the same root as senile or senior. Okay. Of course, cytokinins are used in tissue culture. So cytokinins and auxins are used to induce differentiation of different tissues, because you could get some kind of like non-differentiated stem cells, sort of like a meristem cell. Okay. and use them to differentiate them into roots or leaves. So you can get them from galls, okay? And they prolong the life of stored vegetables. Now the fourth hormone, so we talked about auxins, we talked about gibberellins, we talked about cytokinins. Now we're going to talk about abscisic acid, okay? So in 1949, Torsten Hemberg, okay, found this substance in dormant buds. And what it did is it blocked the effects of auxins, okay? So he called them dormans, okay, because they had a dormant buds that it came from, okay? In 1963, 3 groups of researchers in the USA, in Great Britain, and in New Zealand actually isolated a compound and they named it abscisic acid, okay, in 1967. So they synthesized it in plastids. You remember what plastids are? Plastids are like the chloroplasts, the leucoplasts, the chromoplasts, okay? They're common in fleshy fruits, right? So they kind of help prevent seeds from germinating. They kind of keep them dormant, okay? They inhibit cell growth, okay? And they help leaves to respond to the loss of water, right? Also in drought, during drought, it moves from shoot to root to help preserve root growth, increase root growth, to increase the water uptake, okay? And now the last plant hormone we're going to talk about is ethylene, okay? In 1901, Dmitry Nedjubo, okay, had pea seedlings in a dark lab, okay? What he saw is that their stem elongation was decreased, and they had an increase in stem swelling, and they also showed horizontal growth, okay? When he placed them in fresh air, they grew normally. So what he found out is that the gas lamps that were used to light the lab, okay, were giving off ethylene, okay? So in 1934, argane show that ethylene is produced by fruits. So there you see a green banana growing ever more and more riper, okay, or different phases of ripening banana. Okay, so ethylene are produced by fruits, ripening fruits. They're produced by flowers and seeds and leaves and roots. And the production of ethylene surges after tissues are damaged. So whenever you brush past a plant, and even if you're just brushing past it, that actually damages it a little bit, like bumping into somebody, you know, sort of aggressive, okay? So they saw that in disturbance in a green field, that plants produce fibers and parenchyma, and that cell elongation was inhibited, so you had short plants. And they actually attributed that to the presence of ethylene from the disturbance producing ethylene and producing these effects in the plants. And the presence of ethylene can trigger its own production. So when ethylene is present, flowers fade. So one thing you don't want when you have cut flowers is ethylene present, okay? Another thing is leaves abscise or fall off the plant, okay? Fruits ripen, which you may or may not want, okay? So if you have some green bananas and you want them to ripen faster, what do? You put them in the bag and you tie the bag shut, or you close the bag in some way, right? Because the fruits are producing ethylene gas, so they actually help bananas and mangoes, honeydew, and other melons to ripen, okay? Citrus fruits color up, and you can have apples in the market all year long. You just pick apples before they're ripe, okay? And you'll store them in cool warehouses with low oxygen, so the respiration rate goes down. You can also ripen pear and peaches too, okay? And apples by giving them a whiff of ethylene, and they'll all ripen at the same time, so you can have apples in the market all year round, even though apples tend to ripen in the fall. Okay, you can induce flowering in pineapples with ethylene.